This Week in Virology, the podcast about viruses, the kind that make you sick.
From MicrobeTV, this is TWiV, This Week in Virology, episode 1317, recorded on April 24th, 2026. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It is a beautiful, sunny, bright blue sky, 68 Fahrenheit, 20 C, and just quite a nice day out there. I hope it keeps up.
It's the kind of day when the students say, "Can we have class outside, Professor?"
Yes, they do.
So, well, you figure out how to show the slides. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. We have here 87 and sunny, a little breezy. I know where this is headed. So, whatever, it's a good day.
It's not humid there, is it?
No.
So
of course, I haven't been outside really today. I've been cramming for TWiV.
Yeah, here we are all inside on a very nice day. Well, we can't do TWiV outside. It would be too noisy. Also joining us from Western Massachusetts, Alan Dove.
Good to be here in Western Mass. It's also gorgeous, a little bit cooler. It's 58 Fahrenheit, 14 C, clear blue skies. Very, very nice spring day.
14 here, it's 21 and partly cloudy. It's very nice out. In fact, when I went out before for 10 minutes, it felt really good to breathe fresh air. Mm-hmm. It's really nice.
Yeah, I actually got out for a walk this afternoon after
That's
lunch.
good.
It's very nice.
If you enjoy these science programs, we'd love to have your support. Go to microbe.tv/contribute. Rosenfeld Lab has moved to Philadelphia. They're looking for a postdoc and a technician to work on enteroviruses. They're interested in pathogenesis and immunity, the nature and function of the cross-reactive antibody response, making mouse models for infection, and developing new vaccine delivery platforms. If you're interested, drop her an email. rosenfelda@mlhs.org. Coming up July 27th through 30th in Minneapolis at the University of Minnesota is ASV 2026. Go to asv.org. Click on Learn More below the ASV 2026 logo. We have the link for ASV Care grant application for ASV members for dependent care, and also the link for Kitty Core or Corp.
Kitty Corp. Kitty Corp.
I can't remember.
Just picture a bunch of toddlers sitting around a table in suits, deciding on actions before the board.
Kitty Corp.
Kitty Corp.
Child Care Services sign up. The grant application deadline for those is May 22nd. The organizers, of course, are at the University of Minnesota, Yu Ying Lian, Wei Zhang, and Lou Mansky. Lou is the next president-elect of ASV.
So I guess congratulations and condolences are in order.
You bet. Alan, did you put this news item in here?
I did not, but I just wanted to point out for anybody who if there's anybody out there who this is your first episode of TWiV, ASV is the American Society for Virology. So.
Right.
I'm responsible for
Okay, tell us about it, Rich.
I got this turned into a rabbit hole for me. Okay. So the news item is a Washington Post article entitled "Annual Flu Vaccine No Longer Required for U.S. Military," Hegseth says. So our Secretary of Defense has declared, Pete Hegseth has declared, that flu vaccines will no longer be mandated for the military. As you might imagine, there's a long-standing tradition of mandatory vaccinations for the military in order to maintain a state of readiness Hegseth, this is from the article, said that under the new policy, soldiers would be able to take the vaccine if they believed it was in their best interest, billing it as an effort to, quote, restore freedom and strength to our joint force, unquote. Quote, but we will not force you because your body, your faith, and your convictions are not negotiable, he said. Okay.
Well,
But
wait
we
a
will
minute.
still force you to run out into the line of fire.
Yes.
No, I think that's, we could, you can negotiate.
If
your
we're going to
combat role,
negotiate
I
one of
think.
these
Yeah,
things,
yeah.
we might as well, yeah.
So just a couple of notes as I dove into this. I would note that George Washington mandated variolation, that is the early form of immunization against smallpox, of the Continental Army in 1777, and this contributed substantially to the success of the colonial war effort because smallpox took out a lot of people. There was also, I dug up, there's an article here about the history of mandated vaccines, including a history of the military mandates. There was a case in 1965, United States versus Chadwell, where a military court ruled against 2 Marine Corps members who refused vaccination for smallpox, typhoid, paratyphoid, and influenza, citing religious grounds. And in the court opinion, the quote was, to permit this would be to make the professed doctrines of religious belief superior to military orders, and in effect permit every soldier to become a law unto himself. So this has been military practice and doctrine for 200 years.
More than 200 years.
More
For
than
longer
more-
than we've been a country.
Right. And there you go. The motivation for this, as far as I can tell, is strictly ideological and has no other real practical purpose, and is a bad idea. I was actually thinking about, of all the things that are immunized against, I suppose you could say, well, flu's not all that bad, but flu is bad. Not only that, but it strikes me that if there's a disease that you're going to get and is going to lay you low, it's flu.
Right. I mean, we vaccinate members of the military against a number of different microbes that we don't necessarily vaccinate the general public against, because they are going to be going to all sorts of places around the world where they might be exposed to those things. They're going to be potentially in harm's way of certain things being released, and they're going to live in very close quarters in barracks. And so all of those things make it more important that they get vaccinated. And it's particularly ironic to think about the flu vaccine not being required, given that there was that whole, 1918 Kansas army situation. That does tell us that flu spreading in members of the United States military might be important in some way.
And in every war until the mid 20th century, by far the highest number of casualties has been from disease, not enemy action.
You think Hegseth even knows about how
No,
1918?
he doesn't care. He doesn't care.
know
This is
about any of this
ideological
stuff.
red
No.
meat for the base. That's all this is.
Right.
I think he might actually have been the one who said that he didn't believe in germs. Because he can't see them.
Right.
Well,
So
this
presumably he doesn't believe in gravity either.
he thinks the Earth is flat, probably.
I don't know.
The religion thing, it bothers me because I looked it up. No major world religion excludes vaccination. That includes Christianity, Islam, Judaism, Hinduism, and Buddhism. So I think they're probably afraid. And if that's the case, you probably shouldn't be in the military. But, the variolation that Washington recommended or said they had to do, that had pretty high side effect.
Yeah.
You were, that practice involved taking a little bit of real smallpox from somebody who had the disease and scratching it into the skin of the person that you were trying to immunize. And because that is not the natural route of infection, the outcome, there was less likely to be systemic spread And so instead of a 30% mortality rate, it was more like a 1% mortality rate. But you got this really nasty outbreak of lesions on your arm. You were really sick. It took about 5 weeks to recover. And while that was going on, you're contagious for real smallpox. Okay. So this was not trivial.
And a 1% mortality rate is not trivial by modern standards.
I don't know if you've read, Rich, a book called Pax Americana.
No.
But Pax Americana talks about this in a lot of detail and Revolutionary War movements, and it basically talks about smallpox in early American history, and goes through this in great detail.
Yeah, I should read that. That would be good. One other thing I should note is that there were a couple of during the pandemic, the COVID pandemic, they mandated vaccination with the COVID vaccine, which of course annoyed a lot of people, and several soldiers refused and were discharged. Okay, because it amounts to disobeying a direct order.
Yeah.
Right.
Okay.
It's the way it should be. And healthcare workers who didn't want to get vaccinated, they got fired.
Yeah.
As it should be. Come on, folks. Use your head. Oh, sorry. I forgot. You don't do that. When it's all about ideology, you don't use your head. I saw a quote recently from George Washington, because this is in the news, people are revisiting, and he said, "It seems something has to be done about this," referring to smallpox.
He was probably a, crazy left-wing radical, trying to overthrow the king.
Right.
Really.
Well, I mean, there were other aspects of his life that would certainly not fall into that category.
True.
Like the whole owning people thing that...
Yeah.
Yeah, right,
True.
right.
That was problematic.
Yes.
Okay, on to the literature. We have 2 papers for you today. Pretty neat stuff again. The scientific literature is pretty amazing. First paper is a Nature article. The DNA virome varies with human genes and environments.
And I'm going to read all the authors because there are only 4 of them. It's Nolan Kamitaki, David Tang, Stephen McCarroll, and Po-Ru Lo. And they are from Harvard Medical School, Brigham and Women's Hospital, and the Broad Institute of MIT and Harvard, an all-Boston group.
And as they say in the first sentence, many viruses have adapted to persist in infected humans for life. And if something goes wrong, you can get, well, sometimes it'll be cleared or you can get disease. And so now we have large-scale DNA sequencing of blood from thousands of people, so we've been characterizing the DNA virome. In other words, the population of DNA viruses and also retroviruses within each person's blood for many, many different viruses. And 2 very common taxa are herpes viruses and anelloviruses, which almost sound like Racaniello in a way.
Yeah.
So the herpes viruses, of course, are latent in most people. So they have periods where the genome is silent or almost silent. And the anelloviruses are there and don't seem to do anything. So they're thought to be
correct me if I'm
good.
wrong, but the anelloviruses are a relatively recent discovery, aren't they?
Yeah.
I
so,
was going to say.
They're
Relatively.
not they don't seem to cause a lot of trouble.
yeah,
That's
so
why
the
they're-
trouble.
The anelloviruses are single-strand DNA viruses that have seemed to show up in some of these virome studies. And I'm not sure how much was known about them before that, but it seems like they're everywhere in people at pretty high levels.
It's
Yeah,
kind
I
of like you
think they're
turned on
probably-
the lights in the kitchen at night and all the roaches went running. You're like, whoa, where were all those? Yeah.
I
Yeah,
think,
in fact,
I think they showed up because of the sequencing.
Every
Yeah.
pint of blood has anelloviruses in it because you can't get a pint without it. So, right? And it's harmless, so we don't worry about them. So we know more or less what is in human blood, but what we don't know is how much it varies from individual to individual, across their lifetime, according to sex, and what drives this variation. And so that is what they're doing in this study. They note that when you do genomic DNA studies on people, you can either have blood or saliva is a common one, right? A lot of these services, you spit in a tube and they can sequence it. And so what they did is there is already whole genome sequencing data available. So they just take that data and they can analyze the sequencing reads. And they have 3 different sources. They have 490,000 blood samples in the UK Biobank, that's UKB, 365,918 blood samples, and 49,000 saliva samples in the All of Us data set, and 12,519 saliva samples in the Spark database, which is a database run by the Simons Foundation to better understand autism. It's called Simons Foundation Powering Autism Research for Knowledge, Spark.
So it's almost 1 million samples they're looking at here.
Right. And so they aligned the sequencing reads from all of these sources with sequences of 31 common viruses, 27 DNA viruses, and 4 retroviruses.
Yeah. And so this was basically these were the sequences that they had already determined were not of human origin. So they didn't align to human genomes, and they specifically checked them against 31 known viruses. So this could very much be an underestimate. There could be more viruses whose sequences were not known in this unknown sequence, who maybe someday, if we annotate those in the database, we could find that this was they're also in the human virome.
Yeah. So in this study, they use 2 terms in the following way. So prevalence refers to the proportion of the people in the study who with at least one sequence read pair that aligns with a viral genome. So that's prevalence. And then abundance is the number of such reads from a given biological sample. And so these genomes could be within host cells or in cell-free infectious, they say infectious particles, but we don't know if they're infectious. All you know is that they're particles with DNA in them.
Right. So prevalence reflects how many people have this, and abundance reflects how much of it is in somebody.
Now, what's interesting is viral DNA was infrequently detected in blood data from the 2 databases, UKB and AOU. And most individuals had no sequence read that could be attributable to any virus. That's from blood. And this is really weird, right? Because we know these viruses are near ubiquitous in human populations. But the problem is that there are not a lot of latently infected cells, like of EBV, for example. EBV, 95% seroprevalence in the UK, yet very small numbers of latently infected cells. And so very hard to pick it up.
Right. And those latent cells are probably not in the blood.
Right.
Or, releasing anything that could be detected in the blood.
Right. So a small fraction of blood samples, like here's the number, 675 out of the 490,000 in UKB, had unusually large numbers of parvovirus B19 related reads. These donors had low reticulocyte counts because most likely the virus has infected their red blood cell precursors. So you can associate those levels with the disease.
They've got some great p-values in this paper. That one's the p-value is 10 to the negative 67, which is not the most dramatic p-value in this paper, but they all are. There's a lot that have really dramatic exponents like that.
Yeah.
About 1% of the donors have almost complete human herpes 6A and 6B genomes. And that's because they have integrated into the telomeres of those individuals. About 1% of the population has HHV6A or B integrated into telomeres.
So that's passed down vertically.
Yeah, in every cell. Right? It's in every cell. Now, in contrast, saliva is a different story. There is a lot of sequences, and they were highly prevalent and abundant in the Spark data set. And interestingly, several herpes viruses had bimodal distributions of read counts, right, sequence counts, with saliva tending to have either no reads or many reads. And so the idea is that for these viruses that infect more than 90% of people, the DNA is not originating from constant latent infections, but rather frequent bursts of lytic replication and release. That's why you see either you have it or you don't, right? And for HHV7, these episodes appear to occur continuously, so that the HHV7 DNA was found in all saliva samples from adults. So just think about that, folks. Next time you spit, you got HHV7 in there.
Maybe.
So most of
and if you don't, maybe you'll have it there tomorrow.
Yes. No, HHV7 was continuous as opposed to
that one was continuous,
yeah.
Right? Sorry, you said 7. Yeah.
And throughout most of this paper, they focus on just a couple of viruses, and herpes viruses in particular, and anelloviruses. But I notice in this figure from the saliva, they pick up a number of others that they talk about, or that they at least score. A couple of different human adenoviruses, a papillomavirus, a polyomavirus. That's about it. Cytomegalovirus.
So in contrast to HHV7, EBV DNA is common, but it's intermittent. With half the saliva samples have no EBV DNA and the others do. Now, the DNA from anelloviruses was detected in saliva only a little bit more than in blood, this is consistent with their replication cycle. Some viruses that were rarely detected in blood were detected in saliva at low to moderate prevalence, like Merkel cell polyomavirus was seen. This commonly infects the skin and is seen in 5.7% of saliva samples. In the Spark database, the HHV6A and 6B were found at rates similar to in the UKB study. And what they note is very interesting. Viral DNA load levels were broadly independent of autism spectrum disorder status in the Spark database. So these viruses are not causing autism spectrum disorder. Okay, what about other variables? What is it? This is very interesting what they find here. So because they have so many people, then you could say, are there correlations with age, sex, circadian and seasonal dynamics, right? Age and sex have strong associations with viral DNA load. In the UKB samples, which was mostly 40 to 70-year-old people, the DNA of EBV and anellovirus, an anellovirus called torquetanovirus, was greater in older individuals, whereas viral DNA prevalence was greater in younger individuals for HHV7 and HHV6B. So
again, great p-values here.
Yeah. Yes.
This is 10 to the negative 356, 10 to the negative 451,
I've
10
never
to the
seen
negative
values
200,
like that. It's
and
amazing.
only 10 to the negative 28.
So in other words, the HHV7 and 6B decline with age. So the Spark database and the saliva there, and this has infancy to old age, DNA loads exhibit trajectories that vary with age and across viruses. Most viruses increase rapidly in the first several years of life, and presumably that's the primary infection. And then EBV DNA continues to increase with age, whereas HHV6B decreases sharply from childhood onwards, maybe controlled by the immune system there. The HHV7 read prevalence in saliva increased to more than 95%, and then in middle age it begins to decrease. And HHV7 in saliva decreases in childhood. And the other thing that's very interesting, viral DNA load is consistently higher in men than in women across 7 viruses in both blood and saliva. And that aligns with previous observations of stronger immune responses and lower viral load of some viruses like HIV and Hep B in women relative to men.
Yeah, they only give the one reference there, but I think there's actually a lot of literature about
Absolutely,
a lot of stronger immune responses in women. And they also note that these differences seem to appear in adolescence, which again sort of ties into this being related to some other aspects of sexual dimorphism.
Right.
There was, we've done a couple of other papers recently where there was this sexual bias as well. And that was the response, response to vaccination in terms of measured by dementia. Is that right?
Yes, that's right.
And that was more, the women were more protected than men, correct?
That's right. That's right.
So all of this speaks to the notion that human male and female immune systems are not the same.
Right.
Also, circadian and seasonal variation. This is very interesting. The viral read prevalence increased 1.2 to 1.3 fold from 9 a.m. to 2000 hours. Right? Because they know when the blood samples were collected for both EBV and HHV7. But they had slightly different trajectories during the day. EBV read prevalence was also 1.3-fold higher in winter compared to summer months.
Yeah, that one sort of stood out to me. And I think it's because I've spent some time, going to some conferences where I'm seeing people work on aspects of bat virology, and they spend a lot of time looking at seasonal variation and thinking about, what's going on in terms of, overwintering and hibernation and birthing and things like that. And so I was spending all of this time trying to decide what might be different in some of these months in humans. And I don't really know, but I do know that when people talk about the bat stuff, they're talking about different stressors in their lives. And so I think it would be really interesting to kind of look more specifically at different stressors in people's lives, maybe cortisol levels relative to some of this. I can imagine, for example, if you looked at teaching college faculty members in April, you would find high levels of stress, and you might see that is influencing viral reactivation levels. But I think it's hard for me to figure out what could be going on with month of the year in such a bulk population. But it's pretty clear, especially when you look at the EBV levels, there's a big change there. And so I thought that was pretty fascinating.
I don't know. Winter to me is very stressful.
Maybe that's it.
I mean, it hurts. Winter hurts.
Sure. Summertime, the living is easy, right?
Right. HHV had different and more modest seasonal variation. And so they say the high prevalence of EBV in saliva with similar circadian rhythms as in the blood suggests that lytic events may be really frequent, and this can determine the viral load. Also, the load has very prominent several-fold variation across genetic ancestries, with different viruses enriched in different ancestry groups. Okay, now we can look at human genetics. People are always saying, well, how come my family, only one person got COVID and nobody else did? Well, people are different, folks, and you're going to see that right here. They can do genome-wide association studies on viral DNA load phenotypes. In other words, look at the DNA sequence of the human genome and ask what differences, SNPs, single nucleotide polymorphisms, go with the DNA phenotypes.
Right, because these samples, these almost 1 million samples were collected for genome sequencing and genome profiling efforts. And so not only, just to give a sense of the size of the data set, in case it wasn't clear yet, this is, 900 and some thousand people genomes. And that's where we're finding these viruses, and 31 viruses. And now you can also correlate those viruses with the genomes that they were associated with the individuals.
And obviously, this is a big computational study, right?
Oh, yeah.
Huge.
So, when was the human genome sequenced first?
90-something?
What?
90-something?
Right around the turn of the century, right
Yeah, I was
before.
going to say around 2000.
Yeah.
Yeah, 2000, yep.
And that took, two competing entities.
Yeah.
But lots of people, a lot of money, and a lot of time
Mm-hmm.
to do one sequence. Now it's essentially automated, right?
Well, we've got a quarter century of progress since then.
Yeah. Unbelievable.
If you want to
Just
feel old.
unbelievable. How long is it going to be, or is it, no, it's not really happening yet, before your genome is part of your medical record as a matter of course?
That's a pretty controversial one,
Yeah.
I think, in a lot of places. But I think that if you really wanted to get your genome sequenced just because you could send away and have it done and have it, for an affordable price. I guess it depends on what you think of as affordable. But whether or not then that gets linked to your medical records is a whole sort of bioethical problem.
Mm-hmm.
And the main reason it's a bioethical problem is because of access to care. If the whole world had universal health care, we wouldn't really be having much of a discussion about that because you wouldn't lose your health care coverage for your genome sequence.
Well, there's also some questions of general stigma.
There are some questions of identity and medical privacy, but, a big part of it is fear of losing coverage.
Okay, so they found variation at 82 loci in the human genome associated with viral DNA load of one or more viruses in blood. And for example, 45 loci for EBV, 3 for HHV6B, et cetera, et cetera. And they also found variants associated with DNA load in saliva samples as well. For 6 of the 7 viruses, except HHV6B, the MHC region harbored the strongest associations between common human variants and viral DNA load. Are we surprised, Brianne?
We are so not surprised. That is perhaps the least surprising thing. The only thing about this that's surprising is that one of the 7 did not show up with MHC as the most the strongest association. I would much more want to know about that than the ones where MHC was the strongest association.
Brianne, explain for the lay folks out there why you're not surprised.
So MHC is part of antigen presentation, and it is a protein that's involved in presenting antigens to T cells, but it also is the most variable part of the human genome. So we each have different MHC molecules, and thus we each present different parts of pathogens to our T cells and make slightly different T cell responses. And pretty much anything that has a relationship to the immune system, when you go and look at human genes that are linked to it, you always end up with MHC because it has such a strong role in influencing which parts of a pathogen you respond to.
Okay.
And that variation across the population is a major reason probably that we're still around.
Yep.
Because if you don't have a variable response across your population to a pathogen, then you can have a pathogen come into the population and wipe out the entire species.
Yes. And so there are a couple examples of species at risk because of low MHC diversity.
Too small. Yeah.
But we are fortunate enough that our population, we've seen selection for varied MHC across our population. So somebody has the right MHC for every pathogen.
And
Yeah,
there's no shortage of us.
I was going to say, conversely to Alan's statement, the reason there's so much diversity is that we're swimming in a sea of pathogens. Okay, so there's selection for variation in that to tackle those pathogens that's going on all the time.
That predates humanity, and in the cases Brianne was citing of species that are on the brink, it's because their population has gone down too small, and now they're at a bottleneck and they don't have enough MHC diversity.
Yeah, typically like island populations that are restricted have restricted MHC and they're susceptible to infectious diseases. You know, evolution has done a really good job of trying to protect life on Earth. We're the ones that's screwing it up.
We broke the game.
Anyway, what's interesting is that the associations are unique to each virus. So for EBV in the blood, the top association is HLA, a gene called DRB10404, almost twofold increased prevalence of EBV reads. And they say, well, DRB1 is a co-receptor that EBV binds to enter cells, and maybe that plays some role in it as well. And then they say, as Brianne just said, some of these are likely to reflect the role of HLA in antigen presentation and immune responses. A couple of other examples that are really interesting. So there's a locus on chromosome 5, ERAP1, ERAP2, LN-PEP locus, which encode 3 peptidases that process peptides for MHC display, and they're associated with DNA load of EBV and HHV7. And for HHV7, the association signal is driven by 2 missense variants in ERAP1 that affect substrate preference and enzymatic activity. And for EBV, DNA load associates with variation near ERAP2 and LN-PEP, including a cryptic splice variant that makes a truncated ERAP2 transcript that undergoes nonsense-mediated decay. In other words, you don't get the right protease made, so the peptide's not presented, and the virus is just okay. It's not going to be inhibited. I think that's really cool that they can get it down to that granular level, because it's also based on all the knowledge we have of the MHC system.
It's really cool that they could get into that kind of level of what is actually, biochemically wrong with ERAP or something like that.
And each virus is also associated with other loci, with many genes and roles in viral infection, immunization, proliferation. For example, the B-cell lymphoma, BCL genes, these are associated with EBV DNA load, probably encode alleles that affect survival and proliferation of infected host cells. A couple of other genes, PML, SP110, and MX2, these are HHV DNA load encode nuclear proteins with roles in restricting herpes virus replication. And they also have a couple of genes that influence apoptosis. This is for anelloviruses. It seems that virus-induced apoptosis is important for anelloviruses. There's also changes at the ABO locus, right, which determines the presence of blood group antigen, and which are bound by some viruses. And alleles that produce type A and type B antigens both associate with higher HHV7 DNA load. Well, I don't have either one. I'm an O. Anyway, there are 4 genes in which rare protein coding variants are associated with increased viral load. And these appear to be virus-specific, more interesting sort of things like I just told you. And here's one that's really cool. They decided to look whether GWAS of EBV and HHV7 in participants with African ancestry had any differences. And they found a novel association at a gene that generates a malaria-protective Duffy null phenotype. And that explains 28% and 48% of the African versus European ancestry differences in EBV and HHV7 prevalence. So you get selection for malaria protection, which is huge, and then it also has an effect on viruses. It's very cool.
And isn't Duffy also a carbohydrate on red blood cells? So in some ways it has some similarity to ABO antigens?
Yes, that's correct. Of course, that's where malaria wants to get in initially. All right, now further, a little more analysis on EBV. EBV exists in 2 types, type 1 and type 2, and they wanted to see if there were some differences in the associations there. So the prevalence reads from EBV type 2 compared to type 1 varied significantly by a person's place of birth. Type 2 was more prevalent in individuals born in Africa compared with people born in Europe or North America. And they find antibody responses higher in individuals with type 2 compared with type 1. They say maybe it induces a higher autoimmune response as well. Remember, EBV associated with MS. Also, they find that HLA variation confers type-specific protection against EBV infection when they look at the type 1 and the type 2 data separately. And this is led by MHC allele called class 1B0801. It seems to be protective against type 1 EBV, but it's associated with increased risk for type 2 EBV. So that's really interesting. So depending on the EBV type, the risk is either up or down. They think that maybe this protein has affinity for one or more type 1 EBNA 2 or 3 proteins, but not homologous type 2 proteins. Then they find that EBV DNA load is a risk factor for Hodgkin's lymphoma. So they look for associations between DNA load and there are lots of clinical phenotypes in this 490,000 participant UKB database. And they found 181 associations between viral DNA load and disease phenotypes. Most of them involved higher viral DNA loads in affected individuals, and many are associated with diseases like inflammation or weakened immune function like AIDS, anemia, diabetes, and renal failure. Organ transplantation associated strongly with increased anellovirus DNA load, presumably because you're being immunosuppressed when you get an organ transplant. And also people who were taking immunosuppressive drugs also had elevated EBV DNA loads as well.
The association of EBV with Hodgkin's lymphoma is not entirely novel. People have known that before, and it makes sense because EBV establishes latency in B cells, and B cells are gone nuts are lymphoma. Nevertheless, this association done in this fashion very much supports and substantiates that initial observation.
Right.
It also supports the analysis and the other things that they see because they're seeing sort of known associations.
Right.
What's interesting is that HHV6A or B didn't associate with any clinical phenotype. Again, supporting the idea that these are innocuous viruses, right?
Yeah.
Many of these disease associations with EBV and HHV7 are related to smoking. Cumulative smoking exposure in pack years strongly associates with increased prevalence of EBV DNA, nearly twice as high for the heaviest smokers compared with non-smokers. But smoking exposure is associated with a decreased prevalence of HHV7. Also, cigarettes smoked per day was associated with similar trends, increased EBV, decreased HHV7. So they have smoking has opposite effects on one virus versus the other. That's kind of interesting. And of course, they would like to know what's the directionality here, right? Does the DNA load affect the phenotypes or vice versa, right?
Right.
And so they go through this analysis and they find that the alleles that increase risk for viral DNA load also consistently confer risk for disease, which they say probably the viral DNA is causally upstream of that. They also looked at the relationship between EBV DNA load on autoimmune diseases and blood cancers that are epidemiologically associated with EBV infection.
Right.
For
so
MS,
this is another one that's not especially surprising. People have found correlations for years.
Now, remember, MS EBV infection is a large risk factor, but it's not influenced by DNA load at all. And for SLE and rheumatoid arthritis, also DNA load is not associated with these diseases. But in contrast, all the analyses suggested that the viral DNA load influences the development of Hodgkin's lymphoma. And the idea being that increased DNA in the blood could reflect a higher proportion of B cells, and that may increase the opportunities for oncogenic transformation.
Right. So the concept here is, in the case of Hodgkin's lymphoma, the virus is probably causative. In the case of multiple sclerosis, lupus, etc., the immune system is the problem.
Right.
The virus is the trigger.
It's an autoimmune disease, right?
Yeah.
So that's the study. It's very cool. Major factors that shape the constituents of the virome. Genetics, age, sex, time of day, month of the year, smoking are the big ones.
It's so cool that people can do this.
It's amazing. It's totally amazing. Yes, I love it.
And this is just the beginning,
Totally just the beginning, yes.
Matter of fact, I think, one way to, I've been saying this for a long time, and I think it's still true. Bioinformatics is a good way to make a living.
Sure.
Because we're accumulating so much data and we don't know what to do with it. Okay. So people who understand the biology and are handy with a computer are of real value.
And the math.
Dealing with this, and the math. Yeah.
This is what I keep telling my daughter. Yeah.
Yeah, you have to like bioinformatics, right?
She's a math and computer science double major, so
Well, that's good.
Oh, that'll
right
do
up
it.
her alley.
When I trained, there were no computers. Right? So it helps if they're computers from early years when you're growing up.
Yeah.
Yeah.
Right? Because then you get imbued with it. And I didn't encounter computers until I was a professor at Columbia, right? And by then it's too late.
And you need to, it's best to grow up with computers that you are doing things with other than scrolling.
Oh, yes, for sure.
You're actually, doing some intellectual activity like writing programs.
Yes, totally. Totally. Hey, this is great. And there are a lot of experiments that are suggested by this work, too. I mean, I could see lots of different things you could do. They also say maybe antivirals would be good to try to see if antivirals would have some prophylactic benefit against Hodgkin's lymphoma, given the association, right?
I didn't look in detail at the methods on this, but I presume that it's entirely computational.
Yes, it is.
All the data were there. They just mined the data.
Yeah.
Yeah, they didn't have to do any sequencing. The sequences are all there.
But mining the data is its own challenge.
Oh yeah. It's not like that makes it easy.
But that's why you can have a paper with 4 authors, because if there are 4 people who are sharp on bioinformatics, they can get this kind of thing done.
Yeah, it's really great. I love it.
Yeah.
Okay. Our second paper, equally fascinating. Nature Microbiology, an emerging human eye disease is associated with aquatic virus zoonotic infection.
The 3 co-first authors are Shuang Liu, Di Hu, Tingting Zhu, and the 4 co-senior authors are Linfa Wang, Li Zhenji, Xiaojing Pan, and Qingli Zhang. And author affiliations are at the end of the paper. Nature, you're killing me. You provide a link or something, guys. And I'm scrolling, because it's not really at the end of the paper.
Yeah,
it's just after the notes, right before the supplemental. Here we go. So the team is from the Chinese Academy of Fishery Sciences in Qingdao, Laoshan Laboratory in Qingdao, Qingdao Eye Hospital of Shandong First Medical University, also in Qingdao, Shandong First Medical University, Qingdao. Medical Department of Qingdao University. Don't worry, they are from other places besides Qingdao. I'm getting to it. University of Melbourne in Australia, the Royal Melbourne Hospital, Binzhou Medical University Hospital in Banzhou, China, Ocean University of China in Shenzhen, China CDC in Beijing, Shandong Eye Hospital in Jinan, Qingdao Municipal Hospital, Duke NUS Medical School in Singapore, and Duke NUS Global Health Institute in Singapore.
So Linfa Wang, is that Melbourne?
Yes.
He's at Duke NUS.
No, he's in
Duke
Singapore.
NUS.
Sorry, he's in Singapore, yeah. Okay, so
this
everyone...
is Nature Microbiology just came out.
And it is unfortunately paywalled.
Yeah, I mean, seriously, paywalled. I had to get it from
Yo,
Alan.
I have press access, so for once I was able to provide a paper to somebody else.
My emeritus status gets me most things, but not everything.
Interesting. Well, so as everyone knows that viruses circulating in wild animals, like especially bats, and rodents are zoonotic risk to humans. But what about fish, aquatic animals? Global human fishery activities reached 223 million tons in 2022, supplying 15% of animal protein supply worldwide. It's up to 50% in some Asian and African countries. So this increases human contact with marine wildlife and their viruses, right?
And I'm very pleased to see that Dixon behind Rich is pretty bright for this episode, because he's going to love this.
Yeah, Dixon is loving this.
Yeah, there are so many parts of this paper that I was sort of shaking my head at. And a few places I was sort of like, why did you think to do that experiment?
Yes, I have questions about some of the inspirations here.
But I wouldn't have necessarily thought about fish as major hosts of zoonotic viruses because of the phylogenetic distance between us and fish. I would think about things that are more closely related to us because the virus would need to do sort of less adaptation to infect our cells. And I really liked how they pointed out some of the issues, how they made the point about the issue with contact, because I think that sometimes we forget that the opportunity for spillover is also a big part of this. It's not just phylogenetic similarity. And in fact, because there is so much aquaculture, there is a lot more opportunity for contact. And so I liked being reminded of that here.
Yes.
As you're talking,
ordinarily you think, go ahead.
As you're talking, I'm also thinking that there is enormous diversity in the how restricted viruses are. There are some viruses that are absolutely specific for, say, one species or something like that. And there are others that are quite promiscuous. Not only that, but there are some that are promiscuous as necessarily. Viruses that will replicate in an insect and in a vertebrate because they need to be vectored by the insect. Okay, so anything goes.
Yeah. Yeah. And to Brianne's point about contact, I mean, normally you think, well, fish are in the water, we're on the land, we're pretty isolated, but it's not just Luca Brazzi who has contact with them. It's anybody who's on the fishing boat processing them or at the fish farm,
So the virus in this paper is covert mortality notavirus.
This is such a great name. And it is so named because I was like, wait, why? How can you have covert mortality? So this is a virus that I think was discovered in farmed shrimp, but it actually appears to be a natural virus of Arctic krill, which are another arthropod. So that all makes sense. It ended up in farmed shrimp, and in farmed shrimp, they get this virus, and they're fine as long as the temperature of their tank is closely controlled. If the temperature gets out of control, swings wildly, or goes a little high, then they die from this virus. So it's covert mortality because they're like these dead shrimp walking. I mean, they've got this virus that's going to kill them if the temperature goes up. And as long as it doesn't, everything's fine, but they're kind of, covert mortality.
Yeah, well, as Alan said, first found in aquaculture shrimp, traced to krill, and it turns out this can infect lots of aquatic animals, 20 aquatic species, including echinoderms, crustaceans, and teleosteans.
And just to give an evolutionary perspective of the breadth of that, echinoderms are like ancient, totally unrelated to the crustaceans. That it can infect other crustaceans, okay, yeah. Teleosts, they're vertebrates. That's most fish. That's the ray-finned fish. Like, you go catch a trout or you see your goldfish in the tank. That's a huge category, and they're vertebrates like us. And that's totally different from a starfish.
Yeah, it's amazing. Plus, we're going to see they infect us too.
Yes.
Which is even more remarkable. In fact, the WHO has listed infection with this virus as a emerging disease for animal health. So we worry about that too.
Yes, and we should, even if it can't infect us, we should worry about it because we do enjoy eating food.
Yes.
Next time you crack open that Alaskan king crab, make sure you don't poke yourself.
I'm going out for sushi tonight with my grand.
Well, there's no sharp points there. I think you're okay.
Right.
Anyway, there's the disease in this paper now, another bunch of letters. Persistent ocular hypertension, viral anterior uveitis. Okay?
Yeah.
POVAU. We call it POVAU.
Yes.
So a lot intraocular pressure, doesn't respond to medical intervention. And this has emerged in the past few years, and its incidence is increasing. So the paper here is to try and figure out what's causing it.
And intraocular pressure, high intraocular pressure is what is glaucoma.
Right.
So that causes progressive, vision impairment. And this condition, since it doesn't respond to treatment, is a very serious eye disease that has become, more common in recent years.
Yeah, I wish they had shown data on that increasing prevalence.
Yeah.
Since this is the first I'd heard of this disease.
So many viruses can replicate in the eye. That's called viral anterior uveitis, like herpes simplex, varicella zoster, cytomegalovirus, rubella, EBV. But all these patients with POVAU test negative for those viruses. So there's no correlation between aquaculture production in certain provinces and the POVAU from 2019 to 2021, if you look at the data.
This was when I first started wondering, how
What?
did you actually come to ask this question? Like,
Right.
What?
Well,
it
after you
would not
figure
have
it out,
occurred
I
to me in a
think.
million years to look at aquaculture for an eye disease. Somebody thought this was involved, and I
Yeah.
want to know why.
Well, I mean, in the end, I'm skipping ahead here, but in the end, they pull sequences out of these patients.
Oh, no.
Right,
I
right.
believe the data. I don't know why they chose to
Yeah,
do
but
these.
I mean,
particular- how many
maybe the-
other things did they do that failed before they ended up
Right,
looking
yeah,
at
no,
fish?
As a person who sort of not knowing this and reading the story, you're like, oh, the etiology of the eye disease is unknown. It varied across Chinese provinces. And so we looked at how it related to aquaculture amounts in those provinces. And I was like, oh, what?
What?
Yeah.
Sure. That wouldn't be the first thing I would look at for my correlation.
Wouldn't
but
even
okay.
be the 10,000th thing I would look at, but okay.
So if you then look at 2022 to 2024, you do get a correlation between higher aquaculture production and increased risk. Okay? So what this disease, it presents with recurrent anterior uveitis, right? You keep getting the infection over and over again, and you get then the increased infections. You have increased intraocular pressure and all kinds of other things that happen as well. So the disease is defined as recurrent anterior uveitis with persistent ocular hypertension of unknown cause. So they enrolled 70 patients with POVAU. 82% had more than one anterior uveitis episode. 51% had peak intraocular pressure exceeding 45 millimeters of mercury. They treated them with an
Which
antiviral.
is double the normal.
yeah, that's huge.
That's like twice the threshold.
Yeah.
They treated them with antivirals, steroids, other intraocular lowering medications. 70% of the patients respond for that, but many of them, the rest need basically glaucoma surgery, where they make a little hole in your eye so that the fluid can come out.
Pressure relief valve.
Pressure release. And then during the 2-year follow-up of these patients, 62% had one or more episodes of recurrent uveitis. Okay, so they got some eye tissue from patients with and without POVAU. Transmission electron microscopy, you can see virus particles, 25 nanometer virus particles, which morphologically resemble this CMNV. This is a notavirus, is an icosahedrally ordered plus-stranded RNA virus.
Why did you pick that virus? Again, it's like...
Right. And then we stained with an antibody against this virus.
Yeah.
Really?
Right.
Why'd you pick that
Wait,
one as
hey,
opposed
wait,
to
wait,
all
wait.
the
Time
other
out,
human
time out.
viruses?
Time out. They get peptides, right?
Not yet.
Yeah, not
Later.
yet, but I mean, we don't know what the sequence of discovery actually was.
Right.
Right.
That's right. That's right.
I'm guessing they got peptides at some point in this and said, "Oh, whoa, this is a fish virus,
Yes.
an aquatic
Yeah.
virus."
Yeah,
And that
I think it's
triggered
funny that
everything else.
they didn't
Wow.
present it in that order. They presented it in this order. And it's all, that leaves you sort
the
of
headcanon that
confused.
I came up with here is that they probably got these eye biopsies from these people who are having eye surgery for this new unexplained disease, looked at it under electron microscopy, said, oh, wow, there's a virus in there.
Yeah.
I wonder what that is. Then got, as Rich said, some protein sequence,
Right.
matched it up, said, wow. And then went in with the antibody, confirmed it was this virus, and then went back to the epidemiological
Right. I mean,
data.
how often
Yes.
do you write a paper where the sequence of the presentation is actually the sequence of discovery that
yes.
you went through?
But
Okay?
in this case, it is particularly striking that the sequence of presentation is so out of the blue.
Anyway, so they have antibodies against the virus that react with these particles in the eye sections. They do mass spec. They can see the RNA-dependent RNA polymerase of the virus, and they never see any of this in people without POVAL. So then they can make a PCR assay, and they look at patients with and without POVAL. 100% CNV positivity in patients with the disease and high ocular pressure. And in patients without POVAL, though, they do see 38% positivity for people with just a little elevated intraocular pressure. They also find the virus in the blood, certain number of copies per mil, and it seems to decline as people age.
I need
Yeah.
to send this to my ophthalmologist. You know,
And CMV DNA load was correlated with intraocular pressure. Okay. The sequences from different patients showed 98.96 sequence homology. Well, identity, actually, not homology, with those from aquatic animals. And so these are viruses that look like they came from cultured fish and shrimp. Then they look at antibodies in these patients. They have serum samples from 70 patients with and without POVAL. And the IgM levels are the same in patients with POVAL with the different disease courses. And people with more disease courses had higher IgG concentrations and never see any antibody in the healthy population. So then they say, how are you getting these infections? So they interview people who have POVAL, and so they can find out that among these 70 patients with POVAL, 50% are home-based aquatic animal handlers, 15% are consumers of raw aquatic products, and close contacts of high-risk group, 15%. That's the highest risk for this infection. And so they say unprotected processing of aquatic animals has the highest risk exposure mode, 54%, followed by eating raw aquatic products, 17%, environmental contact, and others, 17%. The incubation time from suspected exposure to the onset is 3 to 12 months, an average of 6. And the incubation period was less than 12 months in 77% of the patients. Among the exposure modes, puncture wounds from aquatic animal appendages, like live or frozen fish or shrimp, pose the highest risk. That's so graphic, isn't it?
Yes.
Puncture wounds. And if they had multiple puncture wounds or ate raw aquatic products on multiple occasions, they have some more severe POVAL and a shorter incubation period. So presumably they're getting more inoculum. Right?
Now, as somebody who's been eating crustaceans all my life, I grew up in Maryland and now I live in Massachusetts. And I've peeled a lot of crustaceans, and usually they're cooked, but if I'm preparing shrimp, they're frozen. They've not been cooked, and you start peeling them, and they're sharp, and this seems like it could be a broader risk than just home-based aquaculture.
Yeah, could be.
Yeah, it also seems odd that, I guess when I first was thinking about this, I was imagining people who were, say, in a fishery working with a really high number of fish and maybe sort of exposure happening through maybe aerosol virus in the area getting to their eye. But here it looks like it's, really the puncture wound from preparing shrimp and then the virus sort of making it to the eye, which is not what I necessarily thought of first. I really, at first, imagined this as something that was, going specifically into the eye, getting, I don't know, splashed by the water or the aerosol or something like that.
But as you know, these other human viruses, herpes and so forth, that get in the eye, they typically do so from the blood. So
True.
It's a good route. So then they looked at aquatic products. They find virus in 54% of fish, 28% of shrimp, 61% of crabs, 58% of mollusks, 47% of cephalopods, and 33% of other species, including spoonworms. And the loads range from 9,000 to 60,000 copies per gram of tissues. And they say if you look at sections of these tissues and you do in situ hybridization, it confirms infection with CMV in major edible tissues.
So now my experiment that I just planned in my head is I'm presuming that all of the fish, shrimp, crabs, mollusks, cephalopods, and other species, including spoonworms, were collected in certain areas in China.
Mm-hmm.
And so I wonder what would happen if I went to the store here in Madison, New Jersey, and got some sushi and did some PCR to see whether or not this virus was present in fish I found in here or fish that someone caught in Massachusetts or, fish in other locations. I wonder how much the prevalence varies.
Although the fish you're getting for sushi is all, I think it's all saltwater oceanic species. Many of them are pelagic species. And so I would expect, tuna in China to be the same tuna that you're going to find in your local sushi place because they've been caught and frozen and air freighted around the world. So I would be interested in seeing the results on that. Yeah. How widely does this travel that way? But also, how widely spread is it in fish farming operations elsewhere in the world and in freshwater fish?
Yeah, this seems like something you could do relatively easily, and now
Yes.
I'm intrigued.
Yeah.
Yeah, it's easy enough experiment. You should try it, Brianne.
I might.
Okay, now they're going to infect mice with this virus. So you have to go to the methods to figure out where they get the virus from. And
I did that, yeah.
They get them from shrimp. They don't actually grow it. They purify it from shrimp. And they say something here that's very interesting. So the isolate was purified from freshly infected P. venomae. And they say, using virus that has been revived in shrimp hosts rather than extracting the virus directly from frozen shrimp samples is critical for successful infection in subsequent cell and mouse challenges. So I guess they get the virus from a shrimp and then they put it into another shrimp to amplify it. Is that what you figured?
You need fresh virus.
Yeah.
Fresh
Guess
shrimp,
they
fresh
don't know
virus.
about what cell lines would work yet.
No,
Yes.
That's what, Vincent and I had the same thought when it comes to they're infecting with virus. Okay, where'd they get the virus? Okay.
Mhm.
Yeah.
And I was wondering, can they culture this or whatever? No. They get it out of shrimp and it's got to be fresh.
So they infect the mice intraocularly. On day 2, they lose appetite. They get increased intraocular pressure, decreased body weight. And on day 28, the IOP is 26% higher than control. IgM levels and IgG levels go up, and they see pathological changes in the cornea, the iris, and the retina, with vacuolization in the iris and retinal photoreceptor and strong virus-positive signals in affected areas. So, holy cow, it replicates in mice.
I would be interested in knowing what happens if you give it to the mice intravenously.
Yes, that would be interesting. Yes.
I would be interested to know if it replicates in cats, because when we started talking about fresh shrimp, I realized that the one place where I come most in contact with fresh shrimp is in feeding my cat.
You feed your cat fresh shrimp?
Sometimes she really likes shrimp.
Huh. Well, you peel it, right?
But they're cooked.
Yeah. Yeah, no, they're cooked. Yeah.
Yeah.
But anyway, it suddenly occurred to me that if it can infect mice and it's in all of these fish products, could it also infect cats? And do we have an unknown cat eye disease epidemic to worry about?
It would be interesting to do serology on cats and humans and some other animals, right? I bet you'd find some seropositive people. Okay, then they do some cell culture. They have human corneal epithelial cells and viro cells, right? Vervet green monkey kidney cells, vervet monkey kidney cells. They infect.
I'm sorry,
with
I just realized what cats you should check.
What's that?
The ones on Tangier Island in Maryland, in Virginia. There's a huge population of feral cats, and there's a shellfish processing. It's like a major center of crabbing in Maryland. That would be my pick for high risk cats. Anyway, continue.
Okay, so they're infecting these.
Good luck getting an intraocular pressure on a cat.
It would be done under anesthesia, I suspect.
So they infect these cells in culture. The cells die, and they look into the cells by electron microscopy. They find intracellular virus particles. So that means the virus can infect mammalian cells from multiple species and cause cellular damage. Now, here's the cool part. They did a survey of 523 aquaculture and wild aquatic animal samples collected from Asia, the Americas, Europe, Antarctica, and Africa. They find it everywhere. See the virus. Highest positivity in Antarctica, 35%, followed by Africa, 20%, the Americas, 16%, and Asia, 16%. Among the 420 samples with identified origins, CMNV positivity was 27% in farmed animals, 32% in wild animals. And the virus from Antarctic krill is phylogenetically closest to the root of the tree when they compare these sequences, so maybe the most ancient lineage. They found CMNV positive samples in 49 species across 10 subphyla of the animal kingdom. And they confirmed infection by histopathology and in situ hybridization in at least 14 aquatic species. Wow.
This is a pretty successful critter.
Yeah.
Very.
This virus goes everywhere.
I wonder.
It's successful, and we first heard of it today.
Right.
Yes.
Well, last week on the stream, someone said, "Have you heard of this fish virus from China?" I said, "No, what are you talking about?" They looked it up and said, "Oh, we have to do this on."
It's not from China. It's from Antarctica.
From Antarctica. Well, the study's from China. They say in the discussion, no virus originating from aquatic animals has been shown to infect humans and directly cause disease up until today. So here it is. That's the first one.
And I was interested, too, in this thing they raised in the discussion, the previous reports that CMNV causes severe damage to eyes and visual dysfunction in fish and shrimp.
Mm-hmm.
This is interesting to me because the eye evolved independently on, 3 different occasions in evolution?
Yeah.
So mollusks and crustaceans, arthropods, and then vertebrates.
Mm-hmm.
And so not only can this virus infect all those lineages, it apparently goes to the eye, these completely evolutionarily distinct eyes in those species. What's up with that?
Yeah, I was wondering actually as you were talking whether they've looked at a bunch of other tissues in animals that carry this virus. Okay?
Yeah.
Is it really eye-tropic, highly specifically, or is it all over the place? And in many cases, the eye is just part of the process.
Which would actually be kind of unusual because the eye is normally isolated, at least in vertebrates. The eye
Immunologically
is
privileged.
immunologically privileged and in kind of an isolated compartment. So it is an odd place to find a virus. It's not an unheard of place, but maybe if the virus can just get into all kinds of different cells, then that's one of the places it goes.
So they make this comment that they have a very low viral load in the blood of patients under 60. So they say maybe the blood is not the best sample to assess infection severity in younger patients. I don't know what else you would use.
Saliva.
Good luck getting,
Saliva.
getting
Yes.
vitreous humor samples from people.
Tears.
Oh, yeah, right. Put a needle in your eye, yeah.
They're going to make everyone very sad and get tears.
Yeah. Tears, yes.
And also in the discussion, they talk about analysis of patients from different regions in China showing that the proportion of POVAU among ophthalmic patients in 6 major aquaculture provinces was 77% higher than the 11 provinces with underdeveloped aquaculture. So also 38% of patients without POVAU but with high intraocular pressure greater than 20 millimeters of mercury were CMNV positive. And they say maybe it establishes a latent infection after the initial exposure, and then you see it later. It's interesting. And so they suggest that people who handle dissected aquatic animals every day without adequate protection or eat raw aquatic animals daily are at the highest risk. So maybe simple preventive measures such as wearing gloves during the processing and handling could markedly reduce. So what are you going to do about eating it raw? Are you going to wear gloves? I don't think
That's
so.
not going to, yeah.
That's a risk factor, right?
Yeah,
Yes.
I mean, I guess when you're eating it raw, I'm thinking about eating sushi.
Mm-hmm.
There aren't a lot of sharp edges.
No.
No. They also have found before that healthy mice that shared water bottles with CMNV infected mice also get infected. Yeah, so close contact may spread the infection as well. It's very interesting. So there's no evidence of human to human spread so far.
Although they did find that there was a distinct subgroup of patients who were students, as they say, young students with uncomplicated lifestyles, had no obvious exposure risk for aquatic animals, but they had family members who had frequent exposure to CMNV.
Yeah, and I think some of the subjects in the paper were positive and were contacts of people who worked in aquaculture.
They also said that during COVID, there was a 107% increase in the incidence of POVAU following SARS-CoV-2 infection, which they suggest may be a consequence of immunosuppression by SARS-CoV-2. So the risk of this seems to be global.
Yeah.
So I'm trying to figure out whether we've seen this before. I'm looking at notavirity on viral zone.
Mm-hmm.
And there are 2 genera. There are alpha notaviruses and beta notaviruses. And the, let me see, which is this? The alpha notaviruses infect insects. Some of these you're familiar with. Black beetle virus.
Yes, right.
Boulara virus, flockhouse virus.
Yep.
Paracodo virus, and let's see here. The beta notaviruses are aquatic species. So we have striped jack nervous necrosis virus, barf and flounder nervous necrosis virus. All of these are nervous necrosis viruses.
Barfin flounder,
Oh,
not
I'm sorry.
barfin flounder.
Sorry.
I heard that too.
The visual picture there.
So I wonder what the symptoms are. A nervous necrosis virus?
I have a new name for a seaside.
I'm glad this is so entertaining.
See, I'm reading about how to extract RNA from fish tissue.
So they noticed that this virus is the second most abundant in the RNA virome of Antarctic krill, up to 73% positivity in samples. And they have previously shown that if you feed farmed shrimp with CMNV positive krill, they get infected. And then they say what Alan mentioned before, that shrimp infected with the virus experience gradual uninterrupted mortality, named running mortality syndrome, when the water temperature exceeds 28 degrees Celsius. So the krill seems to be the reservoir.
So we should look for this in baleen whales, too.
For sure.
They eat a lot of raw krill.
Really a lot. I mean, I saw a number recently that was incredible. How much
Literal
krill?
tons. Yeah.
How much 16 metric tons a day.
Yeah.
Oh my gosh. That's incredible. So they must be pulling this virus in left and right.
Yeah.
So looking up beta notavirus, these nervous necrosis viruses, clinical signs and symptoms can be clinical or subclinical. Signs include abnormal behavior like lethargy, anorexia, spiral swimming, change in pigmentation. Mortalities can be up to 100%, and these have only been known since like 1989, 1990.
Okay. I think that was cool. I like that very much.
Yeah, that was
Very cool.
great. Great discussion.
Okay, we have one email. Brianne, would you like to take that?
Sure. Ben writes, Dear Doctors TWiV, longtime listener, first-time correspondent. My wife and I were wondering how it can be that the MMRs we have from the 1970s are still good when so many others need to be refreshed. Obviously, some viruses mutate quickly, e.g. influenza, but what other factors matter here? You mentioned that the Broad had recently sequenced over 1,000 measles genomes. If the measles virus mutates fast enough for that to be of interest, how are our half-century-old vaccinations still effective, or so our doctors claim? Sincerely, Ben.
It's a great question.
It's
It is a
actually,
great question.
all viruses mutate pretty much the same rate. It's whether the mutations end up changing the antigenicity is what matters. And some viruses can tolerate it and others can't. So poliovirus, it undergoes mutation, but the antigenicity remains the same. There are 3 serotypes, right? And flu? No, it changes every year. I don't think we understand it. Although we did a study years ago on TWiV where they had mutated the gene encoding the entire glycoprotein, and they identified regions that could sustain amino acid changes. And so that's the answer. It's not a great answer, but
Yes, in my mind, the answer is that the ones that are stable serologically, that is, they don't change in terms of antigenicity, are ones where the mutations that would change the antigenicity come at a fitness cost. All right, flu is, my perspective on the flu hemagglutinin is that the real active site of the binding is buried inside this, very decorated thing that has all the antibodies don't actually react with the receptor binding site itself, but spinach around the receptor binding site that can change without changing the activity of the receptor binding protein. Something like that.
Yeah.
Yeah.
The only thing I would point out, at least for MMR, is that the MMR recommendation has changed over time. And so if you got your MMR in the 70s, as you mentioned, Ben, since that time, the MMR recommendation has changed. So it was originally 1 dose. A second dose was added. And so you may, in fact, if you got your vaccine in the 70s, want to get a second dose to be currently considered fully vaccinated. I don't know exactly which year that change happened, but I know that it happened between when I was a child and when I went to college, because I was fully vaccinated as a child and I was not fully vaccinated when I had to fill out paperwork for college.
Yeah, at UF when I was there, they required you to show, actually, they maybe even had you get a measles vaccination, or at least show proof of it before you entered the university.
Yeah, I had to show 2 doses, and I only had 1, and it was very confusing.
Lots of times, in most cases, at least the way we think of it, the second dose is a booster. But lots of times it's also, let's make sure that our coverage is 100%. And the second dose will, get people who actually didn't take the first time or actually never got vaccinated the first time.
So I think it was 1989. Yes, 1989, the second dose was recommended.
That'd be that right time period.
That was around a time of there was an outbreak, right?
Yeah, there was an outbreak then.
That
yes.
resulted. We talked about that just recently.
Yeah, last week somebody asked about it. Yeah.
Right.
Yep. Okay. Send in your emails, folks. We only had 1 this week.
Yeah.
Now let's do some picks of the week. Brianne, what do you have for us?
I have a pick of the week that is a website that I use frequently, and I am always surprised when people have never heard of it and don't understand how incredibly useful this website is. So in some ways here, perhaps I'm giving away my secrets of how I can find out certain information. But this is the Internet Archive. It's usually known as the Wayback Machine. And this is actually a digitization project to digitize and to save old websites. And so you can put in URLs here, and you can get all of the times that those URLs were previously saved and recorded by the Internet Archive, and look at old versions of websites. And so if you want to know what a particular website looked like in 2005, you can put the website into the Internet Archive and actually get the previous version. And so there are lots of times when I've had to think to myself, oh, how did that work back in 1997? And I put it in here and I get the 1997 version of the website. And suddenly I have all sorts of answers about things I definitely don't remember. So this is, as you can see, it's a nonprofit library, and they have videos, and they have software, and they have websites, and they have music, and they have movies. I have used it the most often to find old versions of websites. And again, it is amazing how handy this comes in. And I always am shocked when somebody has never heard of it. So I thought I would do everyone a service. And if they've never heard of it, use the Wayback Machine. It's great.
That's a great pick. And I have donated to them. It's in addition to all the stuff Brianne just said, this is an immensely useful tool for journalists. Not so much for me, but investigative journalists who are often looking at things where the web page has changed in significant ways, and they can find the old version. And just to add one other thing that you can do with this, there is, I don't remember where the link is. It's on this page somewhere. You can add a page to the archive. So if you're looking at a page and maybe you're writing a blog post about it, or you're writing a paper about it, or a legal opinion or something, and you want a permanent link to that version of that page, you can put it into the Wayback Machine, they will archive it, and they'll give you a link to that archived version. It's a tremendous service.
I didn't know about that, but that's really useful.
Yeah.
You can also find websites that are gone, right?
Mm-hmm.
Yes.
If they were archived at some point, you can find them again. Yeah.
Yeah, if you go to if you follow a link, and this comes up in the scientific literature too, so you follow a link to some old article, and now it's like, a parked domain or a spam site or something, and that's not what it was, and you can go to the Wayback Machine and find what it was. Often, if it was archived.
Yeah, so if people don't know about this, I thought they should know about it.
Yeah, definitely. Great pick.
It's cool. Rich, what do you have for us?
I don't know where I got this, but it was in my TWiV Picks box. It's a YouTube channel called Grandparents for Vaccines that consists of 62 short videos that are testimonials from grandparents advocating the use of vaccines. I notice that one of the contributors is Paul Offit. But we often have learned, I think, over time, that the most compelling way to convince people of one thing or another about stuff like vaccines is not necessarily a bunch of data, but anecdotes, stories. And so this is a collection of stories. Some of these people are actually medical professionals or retired medical professionals. Some of them are just grandparents. And I think one of the most interesting things about it is that these people are old enough so that they remember a time before a lot of these vaccines were available. And they know what that was like. I should note that although I picked the YouTube channel, there's actually a webpage called grandparentsforvaccines.org. And you can link to that from the YouTube channel or just look it up. And that has a broader view of the whole thing. So at any rate, if you need to convince some friends that vaccines are good, here's a resource.
This is great.
So that was a listener
That's
pick
awesome.
in TWiV 1305
Oh, is that right?
by
Okay.
Douglas. Yep.
Maybe that's where I got it.
Maybe, because you were on that program.
Was
You were
I on that
show.
program? Holy
were.
cow.
But you
I
can't, it
better
doesn't
go
hurt
get
to
my flu
repeat.
vaccine.
Nope.
Yeah, right. It doesn't hurt to repeat. Alan, what do you have for us?
I have a book pick. So this has been in my list for a while. It is an absolute classic of this topic. It's called On Writing by Stephen King. Even if you haven't heard of the book, you've heard of the author. Stephen King has obviously done a lot of writing, and this is his memoir of that life and of practical advice to the extent that it can be given for writing. Now, obviously, his focus is fiction, but he is, as I realized in reading this book finally got around to reading it a few years ago, he is a fantastic writer of nonfiction, and this is a really, really excellent read, whether you are a writer or just a reader who's interested in the process. A really outstanding book.
He's so prolific.
He is incredibly prolific.
I just don't know where he gets all his ideas
He
from.
he has some interesting takes on the writing craft and the process and life in general.
This has been on my list of books to read for a while, and you just raise it up for right after the semester's done.
He speaks of a horrendous automobile accident or something.
Yeah, he was in fact, I think it was an inspiration for one of his stories, Misery. He talks about it in this book. He frequently goes out for a walk, went out for one of his usual walks and was walking down the road there in Maine near where he lives, and some guy, distracted driver, came around the corner in a van, didn't see him, and plowed into him at a high rate of speed, and he was in the hospital for months, and yeah. But he talks about writing through that process as well, and yeah.
My pick is a YouTube video called "The Real Reason the Soviets Lost the Space Race." Before the Soviet Union fell, we didn't know much about the Soviet space program, but afterwards the records were opened, and this has some eye-opening stuff that is just amazing. So they contrast the American and the Soviet programs, and how the Soviet programs, well, Khrushchev would say there's the anniversary of the revolution on this day, and you have to launch Sputnik on that day. And then you have to launch a man on the next. So there was no long-term plan. So Sputnik, there was no long-term plan. They threw up a satellite with a radio in it, and they didn't have any other plans. And they also didn't test things. So, for example, the Yuri Gagarin, the first guy up in space, they couldn't figure out how to get him back safely. So he ejected from the capsule 7 miles up and parachuted down. Can you believe that? Oh, my God.
I like the bit. I watched this. I like the bit about how they hadn't appropriately thought through or tested the spacesuits for use in a vacuum. So the first guy who did a spacewalk exited the capsule. And since he was in a vacuum, his spacesuit blew up and he was too big to get back in.
Yeah. Yep. Can you imagine that?
I definitely want to watch this. I was talking in the pre-show that I've been watching a show that is actually an alternate history of if the Soviets had won the space race that I've been watching recently, called For All Mankind. And so I think this will go really well with watching that show.
I have to say that my own observation has been, this is just anecdotal, but my observation has been that SpaceX has a high rate of failure.
Yes.
They seem to have a more of a, not quite as bad as what's described here, but more of a Soviet approach to things. Like, well, this seems like it maybe ought to work. And then it blows up and they say, oh, yeah, well, we learned something. And it's an interesting approach, not as sloppy as what's described here, but certainly different than NASA.
SpaceX has a similar situation to the Soviets in that they have an effectively unlimited budget. And they are driven by a desire to get high-profile projects up. They do have the tremendous advantage that a lot of their launches don't have people on them.
Yeah.
So when a rocket blows up, they say, oh, well, it's a few $100 million down the drain. Elon's got plenty more.
I can't believe that they're supposed to land on the moon next year, and they still haven't sorted out how it's going to work between Blue whatever and SpaceX. Come on. I mean, that's crazy.
This is typical of tech bros. Yeah.
Oh, my gosh. Anyway, one more story from this. So one guy is coming back, and his parachutes don't open. And he just slams into the ground. Of course, he was killed. But the whole way down, he was screaming at the Soviet bureaucracy, cursing them for their lack of testing. It's horrible. Oh, and the dog, right? The dog that they sent up, remember that?
Yeah.
Yes.
So Laika was the name.
Yep.
Laika means barker.
Yes.
In Russian. They picked it off the streets of Moscow. It was a stray
mutt. Mm-hmm.
It had 7 days of food and water, and then on the 7th day, it was laced with poison, so it would kill the dog because they couldn't get it back. On day 2, the heating system failed, and it cooked the dog to death. We didn't know about that, right?
Yeah.
Horrible. Anyway, it's really eye-opening. I enjoyed it. We have a listener pick from Ginny. Hello from Lawrence, Kansas. Hey, I was there a couple of weeks ago, where we had a high of 80 Fahrenheit today with late afternoon thunderstorms, and where our weekly local wastewater levels are currently low for influenza and COVID and medium for RSV. I'd like to recommend following Dr. Cal, your science pal, a great science communicator who you can find on social media, including Facebook and Instagram. Dr. Cal has a PhD in genetic medicine and does a wonderful job explaining complex concepts. Many thanks to TWiV, the GOAT in science communication, for the invaluable information you make accessible to all on a weekly basis. I like that. GOAT. TWiV is a GOAT.
Dr. Cal, the science pal, has a YouTube channel as well with 52,000 subscribers.
Is it? So I'm looking for...
Dr. Cal, your science pal.
Yeah, I'm looking And I'm getting something slightly different on Instagram. That's you are Science Pal instead of your.
Yes. Which is this? That looks like the same person.
Yeah, it seems to be the same person.
Yeah, Dr. Cal, your, which is you are Science Pal on
Yeah.
Instagram.
It is the same person, it looks like. So,
Yeah. Cool.
cool.
Yeah, you got more content on Instagram, I think, than on YouTube. Okay, that's TWiV 1317. You can find show notes at microbe.tv/TWiV. Please send us your questions, comments, and pics to TWiV at microbe.tv. And if you enjoy these programs, we'd love your support. microbe.tv/contribute. Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.